right, so today's discussion is going to be about cell membranes. So what cell membranes are made of, and then how we get materials through or in and out of the cell membrane. After today, here are the things that you must know, or the things that you need to be focusing on in today's lecture. So how the membranes and organelles contribute to cell functions. So today we're going to be talking mainly about the membrane and how its structure relates to its function, the major functions of membrane proteins, what makes membranes selectively permeable. We will talk about isotonic, hypertonic, and hypotonic solutions and how water will move based on the solution that a cell or something is in. And then finally, how electrochemical gradients and proton gradients work to transport different molecules in and out of cells. So our first main idea is really about the cell membrane and that they're fluid mosaic models. And so we're going to talk about kind of everything, all the properties of a cell membrane that would contribute to that idea of that fluid mosaic. And so the first thing we got to talk about is, what is the cell membrane made of? And so cell membranes are made of phospholipids. This is something we talked about in Unit 1. A phospholipid is one of the types of lipids that we talked about. But the thing about phospholipids is that they are what we call amphithetic molecules. And so pretty much that's just a fancy way of saying that they have polar and nonpolar parts to them. And so in the case of the cell membrane, the phospholipid is going to have a polar head and two nonpolar tails. So here's our polar part and here's our nonpolar part. So the nonpolar sections are made of fatty acid chains, and then the polar parts are just made of a phosphate group. So what ends up happening with these types of molecules is that they are really good at making a barrier because of the way they want to orientate themselves. And so what you'll find is that when you drop molecules like this into water, they are going to orient themselves so that the polar heads of the phospholipids are touching the water, and then the nonpolar tails will be oriented, to be touching each other in the center. So then you get this area in the middle, which is nonpolar, and then you have your two polar regions on either side. And so that's what the cell membrane is made of, and that's why it orientates itself the way that it does, because of the properties of what it's made of. And so this, orientating of phospholipids is actually something that's energetically favorable. And so here, you could see, you're always going to see the phospholipid bilayer as a cross section, like we see in this top picture, where you have that nonpolar section in the middle and you have the red areas that are polar facing the water. However, that's not the way that material is going to want to stay. And so it is going to, these nonpolar areas are not going to want to be exposed along their edges. And so it's more favorable to make a sealed compartment. And so this is why most cells that we are going to see are going to have some type of, they won't be a perfect circle, but they're going to be enclosed in that way because of that nonpolar region really not wanting to be around any aqueous solutions. Another part of the phospholipid bilayer is cholesterol. And so we know that cholesterol is a steroid. We learned about that in Unit 1. But cholesterol plays a really important role in the cell membrane. And so a lot of times, when we talk about cholesterol and we talk about it in health, we think of it as a bad molecule, right? Something that we don't want a lot of. But actually, cholesterol in moderation is a good thing for the cell membrane. And the reason why is because cholesterol helps to keep these phospholipids kind of in check. So these phospholipids are constantly moving around. They're switching places with each other. You know, all molecules have energy and are going to be moving around. And so what this cholesterol does is it stops these phospholipids from moving apart too much so that, you know, they could create big holes in the cell membrane. But it also, so we don't have big open holes, but we don't want too much cholesterol in there, because if we had a bunch of more cholesterols, you know, interspersed in between these phospholipids, then it would be really hard for things to pass through that, that lipid bilayer. And so, you know, you don't want too much cholesterol, because that would lead to like the hardening of these membrane walls, but you don't want too little cholesterol, because then anything is just going to be able to pass through the cell membrane, and that that permeability, or the choosing of what goes in and out, is lost. So when we think about how we're going to build a semi-permeable membrane, it's not just going to be made of the phospholipids and the cholesterol. There's going to be one other molecule that will sit in the phospholipid bilayer. And so when we think about what kind of properties does this molecule need, we need a molecule I'm not getting that to work. But anyway, we need a molecule that is going to have both polar and nonpolar properties. And so when we think back to our biochemistry unit and we think about the major types of macromolecules we talked about, there's really only one molecule that is going to do that. And that molecule is proteins. And so proteins, if we remember, will have both hydrophobic and hydrophilic regions based on which amino acids they're made of. And so what will happen with these proteins that are embedded in the bilayer is you're going to have regions of amino acids that are nonpolar, and then those regions will orient themselves by the phospholipid tails. And then you're going to have regions with the polar amino acids that will orient themselves towards those phospholipid heads and the aqueous solution that's on the watery parts in and outside the cell. And when we talk about membrane proteins, there's really a couple different ones that we can have. And so some proteins are going to be peripheral proteins, like this one down here. And so peripheral proteins, they are going to be bound to like the surface of the membranes. So they'll be sitting in or outside of the membrane, sometimes connected to another protein or connected to, you know, the cytoskeleton or the matrix or something like that. So that's one type of protein we can have that's, you know, on the membrane. Or we're going to have what we call integral proteins, or transmembrane proteins is another name from them. And this protein right here that's embedded in the phospholipid bilayer would be an example of this. And so these are going to be proteins that span the bilayer. So they're both, you know, their polar regions are on the outside and their nonpolar regions are on the inside. And so when we talk about the kind of functions that membrane proteins provide, these are the kind of the main functions that they are going to do. And so transport is a big one for transmembrane proteins, transport of molecules in and out of the cell. And we'll talk about what type of molecules that happens for in a little bit. When we start talking about cell signaling, we are going to talk way more about the enzymatic activity of proteins that are in the phospholipid bilayer. And a lot of times they're going to be used in sending messages, like within the cell. And then also signal transduction of cell signaling, again, like taking a message that comes from outside the cell and transducing that message inside. They're also going to be vital in intercellular joining. So if like this is cell 1 and this is cell 2 and they need to be connected, proteins are going to provide anchors for that. Also, cell-to-cell recognition. So this is going to be used in things like the immune system. But this is for cells to recognize other cells that, they need to interact with. And so that cell-to-cell recognition can be important. And then finally, just that attachment to the cytoskeleton and the extracellular matrix. So, obviously we're going to have like microtubules and microfilaments that are going to need to come, to the edge of the cell membrane in order to carry vesicles that need to be transported out of the cell. And then also that extracellular matrix, that protection that's outside the cell, is going to need to adhere to proteins to, stay affixed and do that protection. So a couple other things that we will find in the membrane, or kind of on the outside of the membrane, are glycoproteins and glycolipids. And so glycoproteins, this would be an example of a glycoprotein where you have a protein and then you have that carbohydrate kind of connected to the end of it. So a glycoprotein is mostly going to be used for like immune system signaling. So when we think of things like antibodies that are sticking on the outsides of like your red blood cells and things like that, those are glycoproteins, and those are recognition systems, or like signaling systems. And then finally glycolipids, which maybe you'll have like a lipid attached to a carbohydrate, which is an example right there. They're going to a lot of times be used in attachment for cells to cells, and then also for recognition, so that so that a cell can, or a signal or something like that, can recognize what type of cell this is. So then all of this information brings us to the idea of the fluid mosaic model. And so the two scientists, Singer and Nicholson, they proposed, they were the first ones to propose that the proteins were embedded in the bilayer. And so they kind of came up with this idea of like what the bilayer looked like, and they called it a fluid mosaic model. And so the reason we call it fluid is because the phospholipids are always moving. The cholesterol restricts that, but they're always moving. So they're having lateral movement where they're like kind of swapping places. And then sometimes they will flap from one side to the other, kind of switch sides of the membrane, but that doesn't happen as often. So that's what makes the membrane fluid. And then a mosaic is just like a collection of a bunch of different substances that come together in one picture. So like a really good example of this is if like, when you were young, you ever made one of those concrete pads and you put a bunch of different rocks or different, things in it that you thought were cool. That's a mosaic. It's, a bunch of different pieces making up one picture. And that's exactly how the cell membrane is. So we have cholesterol, phospholipids, proteins, glycoproteins, glycolipids. All of these things are coming together in order to make up the cell membrane. Okay, so then that moves us on to idea 2. So we've talked about the structure of the membrane. Now we need to talk about the function and what it does. And so the function of the plasma membrane is to be selectively permeable. That idea and that kind of like vocabulary word is not going away. Selectively permeable just means it's choosy. It decides what goes in and what comes out. And so we're going to talk about exactly how the membrane lets things in and out. So we kind of got to talk about 2 different types of molecules that can be coming in and out of the membrane. The first is nonpolar molecules. And so nonpolar molecules, obviously we've talked about these types of molecules in Unit 1, but we're talking about carbon dioxide, oxygen, little hydrocarbons. These are hydrophobic and can pass straight through the membrane. And so when we think about why can these pass straight through the membrane, well, we have this big region of nonpolarness in the phospholipid tails. So those molecules, because they're small, they can fit through the phospholipids. And when they get to this nonpolar region, they're not going to be afraid of that. They're going to like that. And so that's why they're able to just pass through the membrane. Now, on the other hand, polar molecules, and so when we're thinking about what needs to go in and out of the cell, we're talking about sugars, big, polar molecules, why can they not cross through the membrane as easily? Well, it's because we have this big nonpolar region. And so those polar molecules, even if they're small, they might get to a certain point and then they're going to hit this nonpolar area and they're not going to like that and they're going to bounce back off. And so polar molecules are going to need proteins, specifically transmembrane proteins, to allow them in and out of the cell. Okay, and so when we talk about how things move in and out of the cell, like how do molecules know when to move on in and out of the cell, or how does a cell determine when this is necessary? A lot of this is going to be governed by that second law of thermodynamics. And so the second law of thermodynamics states that, the universe tends towards thermodynamic equilibrium, meaning we want to be equal on both sides. And so like if I took some food coloring and I dropped it into a beaker of water, that drop of food coloring would be really concentrated in one area. And then if I just set it on the counter and I let it go for a while, eventually all the water would be the same color of red or blue or green or whatever color I chose. And that's because of this idea of diffusion, okay? And in diffusion, molecules are gonna move from where they're highly concentrated to where there's a low concentration of those molecules. And so after a while, those molecules of dye are gonna be in equilibrium, meaning we have an equal amount on either side or throughout the entire suspension. So we call this passive transport. And so in passive transport, there is no energy needed. This is something that molecules are just going to do on their own. And so remember, diffusion is moving molecules from high to low concentration. So if we have these purple molecules, they're going to want to move out in this box to where the low concentration is. This is where the high concentration is. Okay, and so again, no energy is needed for this to happen. This is something that will just happen on its own. We see this probably best, or the best example of this is in your lungs and in that CO2 and O2 exchange that happens. And so when you take a deep breath in and you have a bunch of oxygen in your lungs, there's going to be a low concentration of oxygen in these capillary beds in these cells. And so what's going to happen is that oxygen is going to want to diffuse from where it's highly concentrated, kind of in the middle of these alveolar sacs, to where it's a low concentration in the capillary beds, and then that blood that's in those capillary beds becomes oxygenated. Okay, so water kind of we get to talk about it on its own because it's so important. Obviously, we've talked about that a lot in Unit 1. And so now we're going to talk about water and osmosis, or pretty much the diffusion of water across membranes. And so osmosis is just the term for the diffusion of water. And so osmosis occurs when water moves from a high concentration of water to a low concentration of water across some type of membrane. And so I think a lot of times it's just important to, when you're doing any kind of these problems, draw a little picture for yourself if it helps, because that might help you, kind of wrap your head around it. And so here, this YouTube experiment that we see in this picture, this was, it's just a really famous experiment, and it really shows this idea really well. So here, if we have two solutions, one of them, let's say this is like a 1 molar solution of sugar, and this is a 0.5 molar solution of sugar, and they're separated by this semi-permeable membrane right here. Water is going to want to move to where it's a low concentration. Okay, so here on the right side, if we have a 1 molar solution of sugar, that means that the sugar on this side is high. So we have high sugar here, lower sugar here, because this is only a 0.5 molar solution. So if the sugar is high in the 1 molar solution, that means there's less water there. And then in the 0.5 molar solution, there's more water because there's less sugar. And so the water is going to want to move from where there's a high concentration to where it's a low concentration. And so after time, you'll see this water level lower on the left side and increase on the right side. And so that's kind of that idea of that diffusion of water. And so we have, obviously, it's biology. We got to add vocabulary whenever we can. We're going to have a bunch of different words to explain what type of, solution and the direction of osmosis is. And so in a hypertonic solution, there's more solute, less water. So that would have been like the 1 molar solution we were talking about on the previous slide. A hypotonic solution means less solute and more water. So that would have been that 0.5 molar solution we were talking about before. And then in an isotonic solution, we're just talking, it's equal on both sides, equal solute, equal water. And so if we look down at this picture, we have a hypotonic solution on the left, a hypertonic solution on the right. So there's less water on the right, more water on the left. Water is going to move from where it's high to where it's low, and it will move to the right into that hypertonic solution. And so the movement of water in this case is going to go mostly to the right. There might be a very small amount that goes to the left, just because, it's not a perfect process. But most of the water is going to go to the right until we reach that point of where both solutions are isotonic to one another, or we have equal solute and equal water. So when we talk about tonicity, we talk about cells that are suspended in certain solutions. We just always have to remember that water moves from hypotonic to hypertonic solutions. And so based on, whatever cell we're talking about and what type of solution it's in, that's, kind of the level of tonicity we're talking about. And so in an animal cell, if a cell, like this red blood cell, is placed into a hypotonic solution, water is going to come rushing into this red blood cell because there's going to be way more molecules of solute inside this red blood cell than there are outside this red blood cell. In this case, when you pump a bunch of water into a cell, a lot of times it will lyse or it'll break open. And so animal cells in a hypotonic solution oftentimes become lysed. Now, if this red blood cell is placed in a solution with equal number of solutes, then we're saying it's an isotonic solution. And so in this case, water is going to move in and out of this red blood cell at an even pace or an even amount. And so then if we take this red blood cell and we put it into a hypertonic solution, that means there's way more solutes on the outside than there are on the inside. And in this case, all the water is going to move out of the cell and we end up with a shriveled cell. Now, plant cells, we're going to kind of, describe them a little differently because of their cell wall. And so just like the red blood cell, a plant cell that's placed in a hypotonic solution, the water is going to come in. But because of this central vacuole helping with the turgor pressure and because of this cell wall that's surrounding the cell, plant cells don't lyse in a hypotonic solution because that cell wall is keeping that rigidness there for it. Again, just like in a isotonic solution, the water is going to move in and out at the same pace. And then finally, if we put a plant cell into a hypertonic solution, it's going to shrivel. Another word for this that we use is plasmolized. But again, because of these cell walls, the likelihood that, even though there's a bunch of water rushing out of this cell, because of that central vacuole helping with the trigger pressure, the cell is not likely to lyse. And so when you think about plants, think about lettuce, and if, after it sits in your refrigerator for a couple of days, it starts to wilt, the leaves don't look as full anymore, and that's because that's what's happening. The plant is losing water, and so those cells are becoming a little bit plasmalized or shriveled. And so kind of an interesting topic for this, or to talk about, is can you drink too much water? Because we're always thinking about it's important to hydrate ourselves and things like that. And the answer is yes, you can drink too much water. And an interesting story about this was a woman who was competing in a contest at a radio station, and it was called Hold Your Wee for a Wee. And so what these people had to do was they had to drink a bunch of water, like gallons of water, and then they had to be in a contest to see who could hold their pee for the longest. And so this woman, she was competing in this contest, and because she drank such a high concentration of water in such a fast time, she was making her blood and her body this extremely hypotonic solution, meaning that all of the water was rushing into her cells. And so for most of your cells, it isn't a huge problem. You can, blow up some cells and regenerate them, and they're going to be fine. But the cells in your brain, this is not good for, because you have that skull that's there that is pushing against these cells that are blowing up. And so what ended up happening was too much of that water was going into her brain cells, and it eventually killed her because her brain cells were dying at a very extreme rate. Okay, so if we look, here's an example of a problem that you might get. So if we look at this cell is sitting in a solution that is 0.5 molars of sugars. If you add, or 0.05, sorry, if you add the sucrose and the glucose together. And then if you look here in this environment, you have 0.03 molars of sugar. And so in this case, there's more sugar inside the cell than there is outside the cell. So the cell, compared to the beaker, we would say is hypertonic. The beaker, compared to the cell, is hypotonic. So then the water is going to move from where it's highly concentrated, which would be outside, to where it's low concentration. So it's going to move into the cell. All right, the final thing we're going to talk about today is transport proteins, kind of the different types of transport proteins, and how they get things across. And so obviously, we have molecules that can move in and out on their own. Those are small nonpolar. But then for, certain molecules like big polar molecules, signal transduction molecules, things like that, we're going to need a protein. And so those come in a couple different ways. We can have, just transporter proteins or channel proteins that allow diffusion to happen across them. We'll have transporter proteins where a channel has to actually be opened in order for molecules to cross. And then we also have these proteins called aquaporins, and they're going to be essential in transporting water across the membrane. Now, even though water is a polar molecule, it's small. And so it is able to diffuse across the membrane on its own. However, an aquaporin is going to transport water a lot more efficiently and faster. And so aquaporins are only going to passage water, and water can move by itself, but aquaporins just help that to happen at a faster pace. And so one type of protein transport is called facilitated diffusion. And so facilitated diffusion is when proteins allow molecules to pass through the cell membrane, but by diffusion. And so those molecules are moving from that high to low concentration. They're just using a protein channel instead of moving through the membrane on their own. And so specific channels move specific proteins. So that would be like, think of these proteins as a doorway. And so it would be like if every single student in this class had to use a different door to walk in my classroom each day. Each of those doors is for that specific student. Each of these proteins is for a specific molecule or type of molecule. Finally, the last type of transport we have to talk about is active transport, and that's when molecules move against their concentration gradient, meaning they're moving from a low to a high concentration. So this is no longer diffusion. This is moving from an area of low concentration to an area of high concentration. Now, this type of transport is going to require energy in the form of ATP. We haven't talked a ton about ATP yet. We'll talk about it more next unit. But just know it's an energy molecule that cells use. And so the type of protein that's going to do this is called a protein pump. And so that protein pump is going to be activated by ATP to move these molecules against their concentration gradient. The most common example used in biology for this type of transport is the sodium-potassium pump, which is used in nerve cell membranes for action potentials to send messages. And then gated channels. So sometimes protein channels are only going to open in the presence of like some type of signal, so that molecules can only move in and out of the membrane when that signal is present. And this is going to be used in cell communication. We'll talk more about cell communication in our fourth unit. But neurotransmitters use this, voltage gated channels. So like both passive and active transport are going to use these types of gated channels. But the stimulus has to come from that type of communication. So like an ion gated channel, this is where a neurotransmitter would open those sodium-potassium pumps in order to, help send a chemical message. And then we'll also see these stimuluses happen when certain cell communications are happening, and those signal molecules will come, bind a protein, and then cause conformational change, which will send communication throughout the cell. Okay, so just a little summary of what we talked about today. We've talked about 3 different types of transport: diffusion, in which molecules move across on their own, and facilitated diffusion, where they need a doorway or a protein to help them. But both of these are what we call passive transport, where molecules are moving from a high concentration to a low concentration, or we're walking down the stairs. When you think about it, walking down the stairs is kind of more passive than walking up the stairs. So it's easier to move from a high to a low place than a low to a high place. And then we talked about active transport, which is moving molecules from a low concentration to a high concentration. Again, think of like walking up the stairs. That takes more energy in the form of ATP, and so that's active transport. Okay, so the last type of transport we will talk about are endocytosis and exocytosis. And so this is when we're moving like a lot of large molecules into and out of the cell. So remember when we talked about the endomembrane system and how, we might move like a group of proteins that have just been made out of the cell so that they can be used somewhere else? That's an example of exocytosis. And so here, like let's say this vesicle, has a bunch of proteins in it, and these proteins need to be moved out of the cell. This vesicle will move along the cytoskeleton until it comes to the plasma membrane. And then because this is made of lipids, this vesicle is made of lipids, it's just going to be able to fuse with the plasma membrane and then let those proteins come out. But we can also do this bringing molecules in. And so endocytosis is that process of moving them in. A lot of times, like phagocytosis or penocytosis, that's just cellular eating or drinking. And it's just some more terms of endocytosis. But in endocytosis, usually it's just bringing big groups of molecules into the cell. Sometimes these molecules might need to be digested by lysosomes, or sometimes just stuff that's nonspecific that needs to be taken in by the cell. But in both ways, we're going to be moving a lot of matter at once, where it wouldn't be okay to just, use a protein channel or something like that. Okay, so that's it for the cell membrane. If you have any questions, please let me know.